All right, welcome to class. Hopefully you're enjoying your day. I'll be in Denver at a conference, so go and get the notes out that Coach Ackfeld has for you. Go ahead and follow along and take good notes. I'll be back on Monday if you have any questions. And it looks like our extra credit movie will be at 12:30 in the Chris Auditorium. 12:30 on Tuesday after our exam in the Chris Auditorium. Be there or be square. All right, what we're going to look at today is really how do we go from, now that we understand a little bit about the genetic code and DNA, how do we go from that to actually the path of the genetic protein. So the first thing, I've mentioned this a little bit, but some key concepts here is that proteins themselves are not actually produced directly from genes. There has to be an intermediate. It's like the book, DNA is like a book, and it's written in a different language than proteins. So anytime you do that, the first thing you need to do in order to go to a different language is you copy down what was in that book, because remember that book can't leave the nucleus, it has to stay there. And though it's in a different language, you go somewhere else to have it interpreted or translated into the correct language. It's the same way. So in this case, what's going to happen is we're going to take DNA and then we're going to write it down or copy it in the form called RNA and then eventually into proteins. So the individual instructions, when you need to know how to make DNA polymerase, you're going to make a one RNA molecule. You don't recopy the whole DNA, it's wasteful. Because RNA and DNA are used, you got to understand the differences between RNA and DNA. So we're going to go through 4 different things that are the differences between RNA and DNA. First, one of the biggest things is RNA consists of a single strand of nucleotides instead of a double strand. Okay, so it has a single strand, whereas DNA here has a double strand. RNA has a 5-carbon sugar, whereas called ribose, whereas DNA uses deoxyribose. Okay, remember that deoxygenated ribose. So just when you think about that, there's your 5-carbon sugar. These are OH groups, and in deoxyribose, there's just an H, there's no O. That's where the difference is there. One of the major things, the third thing, is that in DNA you have adenine, thymine, guanine, and cytosine. In RNA you have adenine and uracil, guanine, and cytosine. So that's the major difference. These two right here is where you see the difference. The bonding still occurs the same. A goes with T, well now A goes with U, G goes with C, G goes with C. Okay, and the last difference is DNA is only found in the nucleus, where RNA is found in the nucleolus, or the nucleus and the cytoplasm. So the location is the third. Here I'll put all 4 of these points up, let you copy these down. And there they are, the major differences between them as I listed them for you. Once again, the location is a key one. I'll give you here about 15 seconds to kind of fill in your notes. As you continue to copy those notes, I just kind of have these 2 images that kind of will illustrate those 4 points. The first one, that RNA is a single strand of nucleotides. Okay, so here's your single strand, DNA is the double strand. Single strand, double strand. RNA contains ribose versus deoxy. Notice the difference. Ribose nucleic acid there, deoxyribose there. Uracil, okay, is present, where in DNA it's thymine. Uracil's down here. Once again, notice it is still a pyrimidine, okay, a single ring, just like thymine. It's just a little bit different between the two. And DNA's, the location's the last thing that's the difference between them. Another thing that you could say that might be a difference between those is that there's actually different types of RNA. DNA, there's only DNA. You don't have like different types. Well, yeah, there's mitochondrial DNA, but for the most part, it's all the same. RNA, though, there's 3 major forms. In actuality, in my AP bio class, I cover a lot more forms than this. Things called SNRP RNA and all this other SNP and RNA, all this other kind of crazy stuff. But we're going to focus on 3 major types, okay? The 3 major types are the following. The first one, called messenger RNA, which is most commonly called mRNA. That's the easiest way to remember it, messenger RNA. Then we have rRNA, which is ribosomal, and then tRNA. Okay, and they can be abbreviated all three types. When you look at these, all three types of RNA are essentially the processing information for DNA to proteins. Okay, they're responsible for this gene expression. And gene expression happens in two very distinct stages. The first one is called transcription. Okay, so as I was mentioning earlier, when you have this piece of paper, maybe it's the Dead Sea Scrolls. It's in a completely different language. You want to get it. You want to know what it says. The first thing you do, they won't let you take it out of the place it's being preserved. So you got to write it down. You're going to transcribe it. Then you're going to take it to some expert who's going to translate it. And that's the second part. So the last two lines, transcription is when the information in DNA is transferred to mRNA, and translation is when the information in mRNA is used to make proteins. Okay? We call this the genetic code. And so what ends up happening is that we're going to go, this is basically considered what we call the central dogma of DNA, where we go from, of biology, we go from DNA to mRNA. That's an intermediate first, and then from there to the proteins. Okay? When we go from DNA to RNA, we call that transcription, to write down. And then from mRNA to here, it's translation. Okay, and that's the steps. All right. So protein synthesis requires these two steps, transcription and translation. All right, so once again, I drew this again for us. The process of DNA to RNA to proteins is this process of transcription, is to write. That's what literally it means. You write it in this form, RNA, and then you are going to translate it into the language of proteins. Okay. The only thing I would make sure to have on this that's not on there, and once again, I didn't do this diagram, but whoever did, I would make sure to put an M there. What does the M stand for? Messenger RNA. Make sure you have that old diagram. What we're going to do now is look at these two stages in a little bit more detail and kind of go over each one and what's actually occurring. The first one is transcription. We talk about eukaryotes. Transcription occurs in the nucleus. Okay, so it happens in the nucleus, and it begins by an enzyme called RNA polymerase. Now that might sound familiar to you, right? RNA polymerase, because when we talked about DNA replication, we talked about DNA polymerase. And its job was to add bases. Well, now the one that's going to do it to make new RNA is called RNA polymerase. Okay? Here's that second line on there about where it actually begins. So RNA polymerase is going to begin on a location called a promoter. What is a promoter? Okay. The promoter is a specific sequence of DNA that acts as a start signal. Okay. Now, RNA polymerase. Now remember, in DNA replication, we had all these things. We had helicase, we had SSBPs, we had topoisomerase. RNA is much smaller, and RNA polymerase is a jack of all trades. It's going to be able to bind to the promoter, it's going to unwind it, it's going to separate the double helix, and it's going to start copying the bases. So it can do so many things. The last thing that happens, as I kind of said, is it's here the last slide in your notes. So that's basically transcription. We are writing down the genetic code, making RNA from the DNA template strand. Give you a couple seconds to make sure you have all your notes filled in. If any of this part you don't fill in, don't worry about pausing. Don't tell Excel to pause it. I'm going to put this on the portal as well. You can go back, rewatch it, see the parts you need, pause it where you need to. Okay? It's kind of like when I watch the Browns play, right? I pause it in those depressing moments, hang my head in sorrow, and fast forward through their multiple turnovers. All right, enough about the Browns. Transcription, once this occurs, once the DNA is separate, the DNA polymerase moves along the bases, it adds it just like our DNA polymerase, so it does the same thing. Transcription follows the exact same base pairing rules, except now we have uracil, remember. In RNA there's uracil rather than thymine. So now instead of A goes with T as in DNA, U goes with A, okay? The single-stranded RNA kind of dangles out. Behind the RNA polymerase, the two strands reform. We don't have those SSBPs to hold them open. Kind of let you copy these notes. I'm going to add a little image here. So as you kind of keep writing, there's DNA. Notice the DNA polymerase is going to kind of get in right here. Open it up, start coding. Notice it's dangling out. Okay. All of this again, a big thing, transcription. Remember, it's happening in the nucleus. Do not forget that. This is happening in the nucleus. We get this mRNA molecule. The next stage of this process, it's going to have to leave the cell, go out here into the cytoplasm and do the next stage of the process. But before we get done, we need to know, how do I know where to stop? Okay, so that's the next slide. So transcription is going to be happening about 60 nucleotides per second until the RNA polymerase hits a signal that we call a stop signal on the DNA. We call that stop signal, you got it, the terminator. Okay, and once again, if you say that, you better use your best Schwarzenegger voice. The Terminator. Okay, that's the terminator. And what is a terminator? A terminator is a sequence of bases that tells the RNA polymerase to stop adding the nucleotide. And then what happens at this point, the enzyme will detach and release the RNA molecule for the next stage of gene expression. Okay, I'm going to go on the next slide. There are three major types of RNA molecules. Okay, as I mentioned, messenger RNA, ribosomal RNA, and transfer RNA. And whoever named these did a pretty good job because they all make pretty good sense to me. mRNA, or messenger RNA, is a RNA copy of a gene used as a specific blueprint for a protein molecule. When a cell needs a particular protein, what ends up happening is we make a specific mRNA molecule. mRNA molecule gets its name because its job is to carry the message of DNA. It is the messenger of DNA. It's a representation of DNA. Okay. The next one is tRNA, and tRNA is an interpreter molecule, and its job is to translate mRNA into amino acid sequence. Okay. And then last is rRNA, and they get that name ribosomal because it actually plays a role. It's part of the ribosome. It's a functioning unit of the ribosome. And here's kind of the three types of RNA molecules themselves, right? Messenger RNA looks kind of like this down here. Its job is to carry the code from DNA. Notice there's uracil in this place. Ribosomal RNA is down here. It actually makes part of the ribosome itself. And then transfer RNA is this guy here. And we're going to talk a lot about him during translation. So kind of a recap so far of what we talked about. During this process, transcription, during transcription, just the first stage of gene expression, we have taken DNA with the use of the enzyme RNA polymerase to make a strand of mRNA. That messenger RNA is one of the three types, and its job is to carry the genetic information, okay, to the site of translation. We've just copied down exactly as it is one page of the Dead Sea Scrolls. We're now going to take it somewhere to be translated. Remember, all of this so far has happened in the nucleus. So where do we go from here? Okay. Well, we're going to have to copy it, but it's in the wrong language. The language that we're in right now is DNA, and we're going to need it to be in the language of proteins. So what we're going to do is use these codons. We've talked about this already. I've introduced this already. This shouldn't be anything new. Each codon along the mRNA strand corresponds to an amino acid, a start codon, or an amino acid, or signifies a stop signal. One of the things we mentioned already is the genetic code is nearly universal. Okay, all organisms use the same thing. In other words, if I looked at GUC and I looked at it on a sequence of mRNA molecules, on an mRNA, the amino acid valine will be coded for the GUC. Doesn't matter if we're talking about a bacteria, an eagle, a dog, or Joey McCasson's cells. All of them are going to have valine in there whenever you see a GUC. So it's universal. What does this tell us? It tells us we're all closely related, that we had a common descendant from which all organisms live. By the way, we think that's bacteria. And here's this. This will be given to you on the test. I'm not going to expect you to know it. Once again, all you need to do is, it's similar to what we did in DNA, but this is actually more accurate. You find the first letter, okay? So if I give you code for, can't have T, right? This is RNA, rookie mistake. If I gave you U, G, A, all you do is you look for the first letter, that's U, G, okay? And then the third letter is A. That's one of our stop codons. Here, I'm going to give you a couple. I want you kind of on your notes to kind of do these. So here's the code that I want you to use. Oops, let's do this. How about ACU, AAA, GAU, and UUU. Find the corresponding amino acid for each one and go ahead and write those down. I'll give you a minute or two to do that. Do 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 boop 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 boo do 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 boom boom. When we look at this, what ends up happening is translation is going to be making us proteins. Okay, where does this happen? The major thing you need to know is the location. And before the first stage of transcription, it happened inside the cell here in the nucleus. Translation is going to occur here in the ribosome. Okay, this is where the amino acids are going to be floating around in here. Translation happens somewhere else. The mRNA is going to leave the nucleus going to the ribosomes. Okay, and one of the important things is out there is going to be our tRNA molecules, and I'll kind of give you what those are here in a second. Is complementary to one of the 64 codes. Okay, I'll let you copy this down real quick, show you what one of these tRNA molecules actually looks like. So here's the tRNA molecule. Notice here's the specific amino acid that's going to be attached up here. There's the anticodon. Don't worry about all these weird kind of letters in here, but just realize there's 3 loops. Most important one being this one down here.
Were the first ones here?
In this particular case, the tRNA molecule has the codon CCG, and the mRNA strand down here that will correspond to the amino acid, in this case alanine, up here at the top. It's going to attach it to the next one. Another tRNA molecule is going to come in, attach the next. So notice each tRNA molecule will correspond to a specific amino acid.
So there are 3 binding sites that are important. Binding sites are the location where it's going to kind of link in. And so we call those, the sites are known as the E, P, and A site. Okay. The A site is going to be holding the specific amino acid that's next. The P site's the next growing one, and then the E site refers to kind of the exit of this molecule. I'll kind of show you what this looks like here. I'm going to minimize this and then kind of show you. So I'll move that off to the side. There. Continue to write notes. Notice here's the 3 locations. The P site is where the actual new chain is going to be added in. Notice how this is out to the growing chain. The A is like the on deck circle, specific codons that match up. The E is the exit site. So this, I have 3 kind of diagrams here that kind of show this. Once again, here's the P. I always remember as EPA, Environmental Protection Agency. P's in the middle, that's where it's added on. The A site is here, that's the on deck circle. The E is where it's going to move, this part's going to fall off, and it's going to grow onto the growing chain. And you kind of see this here. Each one's, the P is where it's adding, E is where this tRNA molecule is going to fall off, and A is the next one's on deck. Once we get to this point, translation begins when the mRNA molecule binds to the smaller subunit. It's oriented so that the start codon is sitting in the P site. Once the ribosome has been formed, the codon vacates the site. It's ready to receive the next one. So it kind of follows that order that I showed you on the previous slide. Kind of a summary of what's going on here. So the mRNA molecule is bound to the ribosome. The tRNA molecule is going to come in and lay the amino acid, and it's going to follow the codon rules. The amino acids are joined to form that polypeptide chain. And here you can kind of see this, that the strand of DNA is going to determine the mRNA, which then that codon corresponds to a specific anticodon, which is going to get us the actual symbol. So as you can kind of see here, we go through the entire process up here. Here is just that same exact thing. Remember, translation is down here in the cytoplasm. Okay, transcription happens in the DNA. Same concept down here. Okay, transcription happens first, copy the code, then translation. I got a short video that kind of shows this, and then that's the end of the video. Make sure you read 10.1 for homework. Chapter 10.1, we're going to review. I've also put on the portal some review guides for our test, which is going to be the first exam. Thanks. So I'm going to get this video going, and you can watch the rest of the movie.
The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.